Imperial yeast is at it again with their Imperialis project, creating yet another unique proprietary strain through hybridization. In addition to its excellent attenuation and rapid reduction of diacetyl, I-10 Mangostini contributes robust ripe tropical fruit, strawberry, and lychee notes that complement modern hops. And as a Kveik hybrid, it can be fermented anywhere from 78 to 90 degrees Fahrenheit or 26 to 32 degrees Celsius without any negative issues. Head over to imperialyeast.com to learn more about I-10 Mangostini and be sure to pick some up for your next batch of fruity IPA. Welcome to the Brew Lab. We are rapidly entering a future where technology is advancing to help us better understand the whys and the hows that things work. As we learn more about the micro level, we can apply more to the macro level. I'm your host, Cade Job, and today in the lab I'm speaking with Dr. Chris Willig about using gene editing to advance hop research. So up until recently, hops were bred the traditional way by making crosses from parents with desirable brewing attributes. It's long and arduous work, but it's produced the variety in hops that we know and love. Varieties like Sabro, Citra, Mosaic, all bred through traditional conventional practices, but technology like CRISPR and other gene editing techniques have advanced to a level where it can be used to make targeted changes to hop genomes and potentially impact traditional breeding efforts in a positive way. So Chris is part of a research team here at Oregon State University investigating how gene editing techniques might impact hop breeding efforts. And he's on the show today to talk with us about the efforts of their research groups and also some of the ways that this technology might be used to improve things like disease resistance or even going so far as to create your own custom hops. Maybe that's a pie in the sky goal, but we'll certainly ask Chris about it on this episode. But first, I wanted to say another big thank you to our patrons. If you're not currently a patron, I encourage you to head over to patreon.com/brewlosophy and check out the monthly contribution levels. In addition to the feel-good vibes of helping to support the work we do here at Brewlosophy, you get access to a bunch of awesome rewards depending on the different pledge levels that you choose. You can get access to Brewlosophy contributor recipes that we've never published, new discounts each month to YakimaValleyHops.com, and for $3, access to a monthly live Q&A session with a special guest from the brewing industry. We've had guests like John Palmer, Jamil Zainashev, Vinny Chilurzo, and even Mr. Brewlosopher himself, Marshall Schott. And when you sign up, all of these prior sessions become immediately available to you on our private Patreon Facebook page. So please head over to patreon.com/brewlosophy to find all the information and get you access to this awesome resource. Thank you to everyone who's left a rating or review of the show. If you haven't yet left a rating or review, I'd love for you to do so. I really, truly love hearing from y'all. It's a great source of feedback, and it helps others like you find the show. Without those reviews, we wouldn't have the Applying the Science series with Jordan, which is getting rave reviews. I hear from you constantly about how much you guys love the Applying the Science, and it's great. The show wouldn't be what it is today without these reviews. So thank you to those that have left a review, and if you haven't, please take a moment to help me out. Feedback is brought to you by the dedicated crew at Haas, who bred Sabro from a Neo-Mexicanus subspecies growing wild in the mountains of New Mexico. Think you know Sabro? Think again. The monster of a hop is efficient when it comes to delivering big flavor. Use less green to save more green, if you know what I mean. And if you like pina coladas and getting caught in the rain, try it in the Whirlpool. It's full of aroma and chemical compounds that last from the hot side to the final glass, even if you dry hop on top of it. Sabro is known for its incredible blend of coconut, tropical, and stone fruits with a pronounced cream character with flavors of vanilla, cedar, dill, and even mint. Can't decide where to use it? No sweat. Sabro's available in all Haas hop formats to meet your brewing needs, from highly consistent Lupamax pellets to efficient Incognito and traditional T90 pellets. Learn more about this awesome variety as well as the other innovative products Haas has to offer at johnihaas.com. That's John, the letter I, Haas.com. All right, listener Seth wrote in about episode 13, Growing Hops at Home, which is an Applying the Science episode with Jordan Folks. He says, I'm loving the Brew Lab episodes. Having some research background myself, I'm so happy you're giving primary researchers an outlet to share their special skills and knowledge with a wider audience. The Applying the Science episodes are also a lot of fun. Exploring how tightly controlled research may or may not apply to homebrewing is important transmogrification, which is, of course, the Calvin and Hobbes trademark. Got you there, Seth. I enjoy the home hop episode of applying the science, and thought I'd share my process and material stream if for no other reason than to write it down for myself. Now, Seth did share his process, but it's a very, very long read and far too long to read as a feedback episode. So I'm going to read some of the bits of it, and I'll respond to Seth directly in an email. But there were a couple of parts, especially about drying temperature and time, that I thought I'd cover. So Seth says, I have 4 varieties of USDA public source hops selected for my brewing habits and their staggered maturation dates to make picking and processing easier. I live in Minnesota at the 45th parallel and my plants are on the south side of my house and love the sun. When Labor Day's in sight, it's time to begin pinching, picking, and splitting cones to check the lupulin and aroma. I do this by feel and aroma, but I'm amazed to find the picking date for each variety is almost exactly the same each year when I check my records in retrospect. I pick around a 6-gallon pail and then cram as many cones as possible into a food dehydrator. 12 hours at 125 degrees gets me down to about 12% moisture, so I have 2 batches, 6-gallon volume, processed in 24 hours. My basement's wonderfully fragrant during the drying period. Using the data from your episode, I'm willing to move up the temperature to speed processing. On a home scale, I like the higher end of moisture content to allow for better packing in a Ziploc bag for freezer storage. This episode has me encouraged to try higher temps to process faster. The remaining binds make wreaths or are composted. I use my process hops throughout the year for bittering as well as late hop additions. The commercial hops are for aroma and dry hopping. I also have more hops. I always have more hops than I can use or give away, so the leftovers are set out to age for lambic style beers or used as mash hops to aid lautering. I need to explore this in thiolized yeast, but for now I use them in the mash, mostly so I don't have to compost them without living up to their full potential. Also curious about the impact of terroir, I've conducted one trial of home hops versus commercial Northwest hops using Comet. Using the same crop year, I compared YVH Comet versus homegrown in a split batch. Using the Brulosophy semi-blind protocol, I differentiated the samples 12 out of 15 trials for a p-value of 0.01. I liked my homegrown hops best, but will recognize the rampant influence of bias. Thanks for all you do, Seth. Seth, what an awesome feedback. This is just covers so much ground, and it's so great to hear from you and the great experience that you've had with growing hops at home. First thing, I mean, it's amazing that you're picking on the same date each year. And I would say, and I wish I had said it in the episode, but that's pretty common for commercial growers too. I mean, it's a pretty tight window. You're going to be within a few days or at least a week or so most years. Of course, things happen and sometimes growing seasons are a little bit different. And of course, commercial farms are different than home growing farms. All that, caveats apply. But for the most part, hops are perennial and they grow back at the same time each year and for a very short window. Deciding when to pick that like day or, what day am I actually going to pick? It's pretty cool. So you're doing the rub and sniff method too. And it sounds like that's what a lot of brewers are doing at home who grow their own hops. And then of course the dehydration method, 12 hours at 125. Yeah. If you crank that up, you've got an opportunity here for an interesting experiment. You said you're already doing 2 batches. Why not do one at 125 and the other at 135 and tell me if you notice a difference? Now I understand we're talking about this in November, so it's probably going to be September or August of next year before you're actually able to do this. But put it in the back pocket and send me a note next year to see how it goes. All right. Again, thanks, Seth, for this feedback. I'm going to take a quick break, and I'll be back in a few minutes with Dr. Chris Willig talking about how gene editing might be used to help hop breeding efforts. One of the biggest improvements to my brewing practices was the upgrade to stainless steel, and Delta Brewing Systems offers some of the lowest prices on high-quality stainless gear, like the Firm Tank, which holds 8 gallons or 30 liters of wort, comes with a domed lid to reduce the chances of a messy blow-off, and it can hold up to 4 psi of pressure. Delta Brewing Systems also has their own line of brew kettles, as well as one of the lowest-priced all-in-one electric brew systems out there, and their prices are remarkably affordable. If you're in the market for legit stainless gear that won't break the bank, you've got to check out deltabrewingsystems.com. We all know that designing recipes is really fun, and doing it well is so much easier with good software. We at Brulosophy recently made the switch to Brewfather, and honestly, y'all, we could not be happier. Brewfather utilizes the latest technology to bring you the most robust cloud-based recipe design software that can be accessed anywhere, on your phone, tablet, desktop, and even offline. It also works seamlessly with numerous third-party devices to make it easier to monitor every step of your brew. I know change can be difficult, but trust me when I say you need to go to brewfather.app today to try it out for yourself. That's brewfather.app. At this point, you've probably heard of CRISPR and gene editing, or at least genetic engineering. I've had several episodes of the show where we talked about genetically modified yeasts, like, for example, that improve thiol expression or help get rid of diacetyl. And while yeasts have a relatively well-studied genome of approximately 12 million base pairs, which allows for targeted manipulation, hops have a relatively understudied genome, and it's 200 times larger at 2.8 billion base pairs. So thinking about genetically modifying hops is a lot to tackle, but it's doable. As my guest in the lab, Dr. Chris Willig, is here to help us understand. Chris, welcome to the Brew Lab.
Thanks for having me.
So you gave a presentation, which was awesome. It was at the Hop Research Council summer update meeting, and I found it absolutely fascinating. I was, we were talking just a little bit before the show that I feel like people, when they talk about genetically engineering or genetic modifications, there's a lot of buzz around yeast because there's so many of those right now. And I was talking with my wife the other day about how there's all this gene editing and AI, and it feels like we're at this kind of inflection point of humanity, like where we change how we approach science and discovery. And how's that for a big lead in right off the top?
Yeah, I think that's true, especially when it comes to biotechnology. With the, I guess, certain agricultural sectors and consumers of those sectors, there's been kind of a historical resistance to biotechnology in the last, 20 or 30 years. And it seems like some of the people who have been in this field longer kind of anticipate that, but I don't experience that to the degree they do in this day and age. I feel like there's much more of an openness and an excitement about new technology.
Yeah, exactly. I mean, I think so too. Especially, maybe it is the yeast talk that's influenced it, or maybe we're just starting to see more GMO products in the market, right? Like corn and soybean and stuff, like raw agricultural products that are GMO'd now for huge benefit, right? For yield and disease resistance and all these things. And this presentation that you gave at the Hop Research Council was sort of my introduction to like, oh yeah, we could gene edit hops too, right? Like there are things that we could do with hops to help improve breeding efforts or, increase disease resistant fungicides. I mean, who knows? I mean, you're the expert, so I hope to pick your brain a little bit about that. I know you guys have some funding proposals out there and a little bit of research that you're already working on at OSU. And I want to get into all of that, talking about gene editing of hops. But first, I always want to introduce you, the guest. So you are a postdoctoral student with Dr. Steve Strauss at Oregon University. So how did you become interested in studying at Oregon State University? Woo, that was bad. The Oregon State University.
I wasn't sure if I should correct you on that.
Woo, yeah, you should absolutely correct me on that. I'm sorry, go Beavs. How did you become interested in studying biotech?
So I guess at the end of my undergraduate studies, I was in Missouri. Spent my whole life in Missouri up until 2 years ago. I had kind of gone through the junior and senior year crises of what do I want to do. Kind of thought I wanted to be a doctor at one point. Then my senior semester, I decided, no, this isn't for me. So I took a year off, and I had kind of, planned on doing something else besides just getting my initial bachelor's degree in biology, and I got really into kind of the GMO debate or genetic engineering and plants in particular, and became really fascinated with the science and what you could do with it. So I ended up going to graduate school at University of Missouri to study genetic engineering, and mostly in the Midwest and kind of everywhere else. If you're doing genetic engineering, mostly you're focusing on kind of major row crops. So I worked particularly on sorghum, which is a grain crop that's grown to some extent in the U.S. You might know it as milo. And also worked a little bit on corn. And just most of my focus was on developing or just making the techniques to make genetic engineering happen easier to do and increase their efficiency. And then kind of toward the middle of graduate school, my I guess then future father-in-law was getting out of the military, and he wanted to open a brewery, and so he calls me up and he's like, I'm planning on opening a brewery. I know you work with plants. Could you make me a special custom hop variety using your biotechnology skills? And I kind of went, I don't know how feasible that's going to be, but kind of just out of that to humor him, I looked into it a little bit and what it would take, and I found, with a lot of kind of specialty crops like hop, there's kind of a dearth of research when it comes to the techniques you use to do genetic engineering. And just became really fascinated with the kind of unique chemistry and characteristics of hop as a plant. And I decided that I this is something I wanted to pursue. So I got a hold of John Henning at USDA to see if I could, after I was done with graduate school, come to Oregon to work on hops. And, he was the USDA hop breeder, so he seemed like the natural person to contact. But he put me into contact with Steve Strauss, who's a professor of forest biotechnology. So mostly he works on trees. And he has over 30 years of experience kind of developing genetic engineering tools. And so I kind of talked to him, and working with John Henning and Dave Gent, the pathologist for USDA, we wrote a grant proposal together to establish genetic engineering techniques in U.S. hops. And if we got the grant, that was going to fund my postdoc work, and we got it. And so that's what I've been doing for the last 2 years.
Nice. What a cool, what a story, approach over there, right? Like you're studying something totally different, like sorghum, still an agricultural product, but then your father-in-law or their now father-in-law, who was going to open a brewery, says, make me a special hop variety. And you're like, huh. Can I actually do this?
Yeah.
Right? That's kind of what we're going to talk about on this episode. Like, what can we do with respect to hops in terms of gene editing or gene modification? So we'll talk a little bit about how gene editing works, and then talk about some of the challenges and opportunities and how you've worked to optimize the technology. And that's kind of been the focus of your research. So we're going to spend some time there and dig down into the weeds a little bit. And then we'll take a break and we'll talk about some examples of hop biotech research. So there's one project that you're currently working on at OSU, but then there's a bunch of other proposals that you have out there. So this is like research that could happen in the very near future, assuming it's funded. And then we'll finish up talking about, hey, what are some other things? Where's this technology going in the future? And then that age-old debate, right? Can you use GMO hops in the marketplace? That seems to be one that people love to talk about. And of course, we had an episode on that with Laura Burns, Molly Browning, and Avi Shayevitz talking about that aspect for yeast. So this will be an interesting sort of, follow-up to that discussion. Much shorter, but but also in the same realm. So cool. All right. So we have to start at the top. I have heard genetic engineering, genetic modification, and gene editing. So what is gene editing to you?
So these are kind of notoriously confusing terms where people kind of use them interchangeably and overlapping. I guess I would start with genetic modification. It's genetic modification, or GMO, is more of kind of a vernacular term that people tend to use that doesn't have a lot of, I guess, clear boundaries of definition to it. So I try to avoid using it as much in science, although it's, sometimes necessary to use, because people may not know like what may have never heard the term genetic engineering. So like, oh, GMOs. Oh yeah. I know about GMOs.
Yeah.
So genetic engineering, I would say, is a more specific term that we use in plant science, or I guess just biology in general, to say that we're taking genetic material that can originate from anywhere. It can originate from an organism completely different from the organism you're trying to put it into, or it can originate from the same organism. Like say you want to move a trait from yeast into a different yeast strain without having to do mating. You essentially copy genetic material. You build what we sometimes call a construct, or just kind of a blueprint of that genetic material, and then use some method of delivering that into the organism's cell, where it then can uptake that and incorporate it into its genome. And as it replicates and produces offspring, it will pass down that new trait to its offspring. Now, gene editing is, again, kind of overlapping with genetic engineering, but it allows you kind of a unique degree of freedom that you wouldn't have otherwise, where the goal is normally to modify a gene that is already in the genome of the organism. And so what you're actually doing is delivering tools into the cell, or blueprints for those tools. The cell then builds the tools, will modify a gene at a different place that you kind of choose as the engineer. And the change that takes place is essentially equivalent to changes that happen in nature all the time that we make use of for breeding and improving crops.
Uh-huh. Interesting. So that's kind of like it's like you go to a builder, and the builder's got a set of blueprints for a house that they want to build, right? And you say, no, use these blueprints. Here, use these blueprints, and then they build like the house that you want them to build, right? Like it kind of like that. Is that what you're talking about? But I mean,
that would be the precision element versus breeding, which I guess would just be you have two houses with a lot of elements, and you kind of so, have like a blind build and kind of see what kind of mixtures of the two elements you get.
Right. Breeding is you go tell two builders to build one house, right? Go and build one house, and that's it. That's the only instructions you give them, is go build a house, and you see what happens.
Yep.
Cool. All right. So gene editing. All right. So I think I understand that then. So gene editing is very targeted, right? I like that blueprint example. So you're giving the yeast cell new blueprints to follow, and telling it, hey, use these genes, not these other genes, or whatever, that's already sort of in the structure. Now, I have to ask then about like CRISPR and Cas9. So like cutting things out of the genome and moving things around. Is that also gene editing, or is that more like genetic engineering?
So CRISPR-Cas9 would be a tool that we use to accomplish gene editing.
I see.
So gene editing did exist before CRISPR-Cas9 was around, but it was the tools that we used were very difficult, not very user-friendly, expensive, and therefore it just wasn't that widespread. CRISPR-Cas9 is very customizable. So what it essentially consists of is you have a protein called Cas9 that performs a cutting function on DNA. And you have another component which is called your guide RNA, which I often analogize this to like a homing device or an address that matches to a specific spot in the genome with just 20 or so DNA letters. You have that guide RNA and Cas9 protein make a complex, so they become one thing together. And then you have that guide RNA. Once you get these components inside of a cell, the guide RNA helps traffic the cutting component to the specific spot you want to edit, and then it will make a break in the DNA. And there are actually several iterations of this. Instead of we may think of CRISPR-Cas9 as like scissors cutting DNA in spots. There are modifications that you can make to the system where you completely turn off the cutting function and have a different enzyme fused to it that just changes one letter precisely. So people will sometimes analogize this to like, well, instead of scissors, we have a whole toolkit now. But this kind of basic application where you use the scissors, you cause this break in the DNA. And this is something that happens all the time in cells in nature. So cells have pathways to repair that cut that you make. And the goal usually with gene editing is to get just a difference made in that repair that causes a trait change in the organism based on that specific gene that you targeted with CRISPR-Cas9.
Yeah. Interesting. So okay, again, yeah. So it's very targeted. You're going in and using scissors in the basic application, but I understand what you mean when you're talking about you don't have to just cut. You can make specific base pair changes and other sorts of edits to the genome without actually having to like break the DNA chain. And that's kind of where, as this technology grows, we're going to figure out more and more and more uses of it, right? For CRISPR and Cas9.
Yep.
Yeah. And what you're sort of describing is the very basic. And that's kind of, I think, probably the easiest one for people to understand, right? Is like cutting the genome in one place and either inserting something in or taking something out. And then, like you said, the whole point is that changing that DNA causes a trait change in the organism that we can observe, right? That we see. And so you mentioned during this presentation, there were kind of two power applications for this technology, or for gene editing, and you called it basic research and then like direct improvement of an agronomic trait. So tell me about those two and what you mean by those two applications of this gene editing technology.
Yeah, of course. So I think when most people think about gene editing, or we can say genetic engineering, or ways of biotechnology that existed before gene editing was a common practice, people would think of this as, how is this going to be used in agriculture? How is this going to work in the field? Are consumers going to accept it? So that would be what I'm calling the actual application in the field. But I think what's really underrated by that is just the powerful use of CRISPR-Cas9 as just a pure research tool. Say we're not trying to make something necessarily a product to put in the field. We just want to understand what gene controls this trait, what gene controls disease resistance in hop with respect to powdery mildew, or what gene might control the height of the plant. CRISPR really provides kind of a unique way to examine this, because especially with the hop genome being so large, that kind of limits the tools that we have traditionally used in genetics to identify genes that are linked with traits. But with CRISPR, what we can do is say, okay, I have a genome sequence of my hop. I have at least a set of candidate genes that I think might be associated with this trait. I can go in and now turn them off one by one, and then look at the offspring that I've altered, grow them in a greenhouse, and see, did that affect my trait? And then maybe among that pool, we identify, one gene, hopefully, that does strongly affect that trait, and we can say, there it is. And that has a lot of implications for just traditional breeding. Say you don't even have to say, we're going to edit something and use it in the field at that point. You could say, we've identified this gene. Now we know exactly what gene it is, so we can, cross a line that has a version of that gene that confers a better trait with one that has other good agronomic traits, and then we can track to make sure that the better version of that specific gene made it into our offspring that we want to look at when we're breeding for new hop varieties.
I see. So there is kind of this overlap between those two in a way, right? Like, or kind of like a circle in a way, right? Like if you want to improve the specific trait, like let's say, for example, hop creep. I think this is a good example for us because Matt Cottrell recently discovered genes in hops that are responsible for amylase activity, right? And so that amylase activity might be what's contributing to hop creep. So say we identified these genes, right? That's kind of like the basic research. We identified some genes that might be related to hop creep. We go in and turn those genes off, and then we see if those same hops creep, right? Whenever we put them into beer. That's kind of a basic research, but also a direct improvement of like the field application. If that's right, then you've now created a variety that's not creeping anymore, right? Which is a trait that I think some brewers are going to like. And so I think that's kind of where it seems to me that's kind of a circle of this application, or at least how these two things are interrelated. But then you tell me the situation like using basic research to supplement breeding efforts, right? Let's say, okay, we've identified this, we've done our basic research, we've turned this hop creep gene on and off. And now we know that that gene is the one that's responsible for hop creep. So we can say, okay, let's go and identify some of our breeding stock that has those genes that doesn't have hop creep and see if we can breed them in, right? Like Cascade is one that's got high hop creep, at least in research that's coming out of Oregon State. The Cascade has high hop creep. So what if you could take Cascade and breed some lines together, or find a mutation of Cascade that doesn't have hop creep, and then start breeding that out and eventually breed hop creep totally out of that variety. That's kind of where you're talking about, like that basic research can be super powerful and fundamental and help not just gene editing techniques, but give breeders something that they can target in their breeding efforts.
Yeah, I think that you got it exactly right. And I think that's a really good example to use here.
Ah, very cool. Yeah, and that was actually interesting too. So I just talked to Dr. Steve Strauss, or well, I saw the presentation at the recent MBAA conference, which is in Seattle, just recently, and he was describing that as well too. He was kind of saying like, gene editing is very targeted. You can specifically identify traits, or you can do some basic research, whereas breeding can produce like some more drastic changes. And so it's kind of like, but they don't have to be separate and apart, right? It's not like you have to do just gene editing or GMO or just breeding. You can use these two together.
Yes, they're very complementary. Gene editing allows you to do things that you kind of can't with conventional breeding, but also conventional breeding has many, many advantages over gene editing that you wouldn't be able to do with gene editing. One really big one is with breeding. I guess the process itself is made much simpler, and you have to do less screening for stuff when you have a sequenced genome. But fundamentally, you don't need a sequenced genome to successfully breed for a specific trait. Really, all you need is to look for those traits and have a good way to kind of screen for them. Whereas gene editing, we really have to know something about the sequence starting out before it's of any use to us at all. And the work it takes to kind of link genes with traits, what specific region of the genome causes this trait out of the whatever 50,000 genes that are in hop, that's an immense amount of work. And if you just don't have the data for that yet, the only way we have to get there right now is conventional breeding.
Yeah, and I mean, this is fascinating too, because I think I just did this episode with Laura Burns talking about the HCY1, the hazy gene in yeast, where she did a combination of traditional techniques and CRISPR. So they saw a phenotype reaction, right? They saw that putting different types of yeast in beer caused some beers to be hazy and some beers to not be hazy. So they start asking why, start looking into the yeast genome, and they actually did back crossing, which is kind of a traditional technique. Find a gene or back cross into a wine strain or whatever, then find a targeted gene, sequence the genome, and then turn that gene on and off in regular yeast, and ta-da, they've identified the hazy 1 gene. It's a huge amount of work, but it's super, super useful and super rewarding when you can actually find something that causes a phenotypic change. And one more comment real quick too, before we move on to how this works, is that, I'm probably going to get some flack or some feedback by this, but it sounds like as humans, we've been doing gene editing for thousands of years, right? We're just not as targeted as we can be now. Now we have some really, really targeted tools to do some gene editing, like breeding itself, like for example, dogs or domesticating, cereal crops and stuff could be considered a very, very, very primitive form of gene editing, or maybe that's too far.
Well, I guess I would amend that to say, maybe we've been genetically modifying things for that long. So like I said, genetic modification is not a super precise term, but if you kind of take it at face value, influencing the expression and kind of distribution of genes within an organism's population, yeah, that's something that we have been doing forever. Conventional breeding is a form of that, and even before agriculture existed, people were kind of unconsciously doing this, right?
Totally.
With,
Yeah.
the domestication of cats.
Yeah. Yeah. Right. Exactly. Or dogs or anything. We've been sort of unconsciously doing this for 1,000 years. And so DNA can be very, very, very large. Right. And as I said earlier, hops have 2.8 billion base pairs per chromosome and may have several chromosomes. So how do you figure out where to make these changes? Like, how do you go through? It's like, surely you're not just sitting there going like CAT GC, and like writing it down on a piece of paper and then like posting it on the wall or something and trying to figure out how to figure out where you should make these cuts. How does that actually work?
Yeah, so that's right. We don't do this manually at all, and it would be very, very difficult to. So I guess the hop genome is 2.8 billion base pairs total, but that includes 10 chromosomes, 2 copies of each chromosome, and then hop is dioecious, meaning that there's boys and girls.
Oh, jeez.
And so they have a separate. They have like a Y chromosome, basically, all the same system as humans.
Yeah, sex chromosome. Yeah. Mm-hmm.
So yeah, it's a very large genome for plant species. So cannabis is the kind of closest, I guess, commercially viable relative of hops. So a lot of when we're looking for specific genes, we can kind of use what is known about other plants to kind of infer that the function of this particular gene that shows up in our genome sequence and hop, it might have a similar function, and we can kind of search the genome for related genes from other organisms that have been confirmed to perform a certain function. I guess what actually is absolutely essential here is in order to do genome editing, you almost have to have a sequenced genome, something that you have like a good percentage of the total genes that are being at least turned on in that organism at any given time. And until recently, we really didn't have a good version of the hop genome. The first one, it was the first one of really a lot of value was in Cade, which was published only in 2020.
Whoa.
So it's been pretty recently that this has been kind of viable. So I guess I'll give an example of the project that we originally proposed to do with the project that I've been working on for the last couple of years, and that was to identify a resistance gene among a kind of set of candidate genes. And so particularly resistance genes for fungal pathogens, they have a certain pattern to them. You kind of know when you look at a genome sequence that, oh, this is a resistance gene or R gene. And so we were able to identify within this region that had been kind of mapped by more conventional genetics of saying this chunk of the genome somewhere in here is this resistance gene. We took that region and we looked at like, okay, what are the genes that fall in this region that actually look like resistance genes based on other plants? We identified several of them, and now those are candidate genes that we are going to try to target with CRISPR and knock them out individually and then test them with, I guess, what we call phenotyping, looking at does the trait actually express here or not. We screen these single edited ones and can identify a specific gene that will have the phenotype eliminated.
I see. And I'm sorry, I'm like taking it in because I'm sitting here. I had a question that I was going to ask, and then I changed my mind to ask a different question. So it's kind of like you're looking at you're not just limited to just the hop genome. What you have to have the hop genome sequenced, right? That's as I understand, that's kind of like the starting point. You have to have it sequenced, but then you're able to look at, for example, resistance genes in other organisms, and or in where you think it might be in hops, and say like, okay, resistance genes take this sort of pattern. Can we find this pattern in the hop genome sequence, and then can we start to mess with some of those genes, right? Can we start to play with some of those and use CRISPR and Cas9 to target those, and then see if we get a phenotypic change, like a trait change in the organism?
Yeah, that's essentially the case. So I would say one thing you need a whole genome sequence for as well is you want to make sure that you don't kind of accidentally edit something that you weren't intending to. So a lot of times, so I guess to get into maybe a little bit of technical detail, the guide RNAs, so like the things that perform the homing function to provide the specificity for gene editing, there are programs that will take just genome sequences input that will help you design a guide RNA that will not only hopefully efficiently cut the gene that you want, but also make sure that it doesn't cut elsewhere in an unintended way, which in that case that it would kind of confound your phenotyping results, because you couldn't say for sure whether a trait change was caused by the thing you intended to target or what we call off target in that case. So to prevent off targets, we really do need a fully sequenced genome, like the Cascade build.
Yeah, and as I'm sitting here, I'm starting to think too, like, wow, what an application for potentially AI to come and help too, right? Like as like figure out where you can cut, what you can cut, and then so you can minimize off targeting. It sounds like you've already got some programs out there that are helping with the RNA. I mean, this could be an opportunity for improvement or using things in the future. But we are pushing up against a quick break, and I hate to just throw the AI word out there without getting a comment on it from you, but we'll do it after the break. So we'll be back. We're going to finish talking about how to edit genes, and then talk about some of the examples of gene editing that OSU has planned and proposed, and even a project that they're working on currently. So we'll be right back.
Established in 1995, More Beer has been consistently serving the greater brewing community since the times IPA was expected to be bitter and clear, and there are reasons they've stuck around so long. In addition to their massive product selection and excellent customer service, More Beer has locations on both the west and the east coast of the United States, which translates into fast shipping times regardless of where you live. And when you spend more than $59, shipping is free. When you're in need of brewing ingredients and gear, there's no better option than morebeer.com, one of the most trusted shops on the planet.
CRISPR-Cas9 and other gene editing tools are becoming household names as genetic engineering works to help advance agronomics in not just the brewing and hop growing and barley industries, but in many other agricultural industries. It's pretty cool technology, but right now it's a little limited, which is okay as things start to grow. Chris, one of the questions that I had was, it sounds like gene editing could be really fast, right? Like you could just snip a gene and then see very quickly what the genotype effect or the trait effect would be. But how long does gene editing really take?
Well, that's a kind of a complicated question, but if we're just talking about laboratory research here, I guess even then it depends heavily on the specific trait that you're wanting to measure and how what tools you have for phenotyping. But we'll take the case of disease resistance. So what we're actually doing when we genetically alter a plant organism, most of the time we're using tissue culture. So this is a kind of synthetic environment for the plant where you want to make sure it's happy and gets all the nutrients it would kind of in the field. but you want to be able to kind of control its growth. So we first use an organism called Agrobacterium tumefaciens, which kind of acts as a middleman. So we'll deliver our genome editing blueprints to Agrobacterium, and then Agrobacterium has this natural method of delivering genes into plant cells, almost by kind of a micro injection process. So we'll take plant tissue, expose them to Agrobacterium that carries the blueprint for the trait we want. We'll put those on plates, and then the idea is to get specific cells that have had the new gene delivered to them to regenerate into a completely independently growing plant. So in hop, with our research so far, it's taken about 6 weeks from exposure to Agrobacterium until we can get an identifiable new hop shoot. And then we will kind of baby that shoot, kind of nurse it and put it into new media, make sure it's really happy, and just try to get it to survive and elongate. And eventually we want to, get it to produce roots. We'll then transfer it out of tissue culture we'll pot it with some soil. And then you have, a small plant that can reproduce just like a cutting that you would take in the greenhouse outside of tissue culture. So for disease resistance screening, we essentially want to get the plants big enough where we can remove individual leaves from them, inoculate them with a strain of powdery mildew, and see how well it colonizes the leaf. That's how we measure resistance. And so we can actually do that when the plant's still pretty small. So about 3 months or so from initial taking plants in tissue culture and delivering genes to them to actually screening for our disease resistance, and then seeing whether the modification worked.
Jesus, that seems incredibly fast, right? 6 months. I mean, 6 months is a long time. Don't get me wrong. That's not like there's a lot of time in that area. But as I understand, traditional breeding efforts are like way longer. Like if you were just going to take 2 plants and breed them together to increase the same trait that you're looking at, that might take years.
Well, so I think the way conventional breeding works in hop, first of all, it's a little bit different than a lot of crop plants, because in corn, for instance, you might want to, the kind of conventional approach is we have this line that has all these great agronomic qualities, but it's missing this one trait that makes it, I don't know, let's say sweet corn or something. And so you'll take a line that has that trait for sweet corn, but the rest of its genome is not favorable agronomic traits, and you'll cross those. You'll select seeds from the offspring that have your sweet trait now, and then you'll just do what's called back crossing back to the parent with all the good agronomic traits. Take seeds from those, repeat for 5 or 6 generations until you get a plant with a genome that is almost completely the good agronomic parent, but just has hopefully the trait added that you wanted. With hop, you can't really do that well because they're very intolerant of inbreeding. They really don't like kind of same family genetics to be replicated over and over.
Okay.
So what you essentially do with hops is you take, a mother and father that hopefully you've identified have carry traits or carry genes for positive traits. You make crosses with them, you get a whole bunch of offspring, and then you kind of gradually select for those offspring that have important traits. Primary these days might be disease resistance, so that might be one of the first things you screen for. And then, you're going to want to grow them up to where they produce cones, and you can do pilot brews, and there are several steps of elimination that you're kind of taking them out of the pool before then. And by the time you get to something where I have a new variety with a good agronomic traits, growers are going to want to grow it, brewers are going to want to use this, it may be 10 to 12 years from the initial cross that was made in the first place. So I wouldn't say necessarily gene editing is faster when it comes to I guess it all depends on whether you're using it to make a product or just to test a trait.
Yeah. That makes sense. Like where you're coming at it in the process, right? Like, are you coming at it in the process from like, I'm like your father-in-law wanted, right? Like I want go make me a new hop, right?
Yeah.
That's a very long-term trait because you're still going to have to do all of the things that a traditional breeder would have to do in screening for all of the other things, not just powder, or, great aroma characteristics or great brewing characteristics, but great agronomic characteristics. And so all of the other things that make a plant complicated. So that makes sense. But on the other side too, you might be able to help at the end if they've got like a really good, really promising strain and it smells great and it's got all of this, but it's susceptible in this one area to this one specific disease or whatever. That might be an opportunity to target something with gene editing and say, okay, let's see if we can take this plant and turn that gene off and see what happens, right? Then You're not like restarting the wheel.
Or you could do it the other way. You could do it where we don't have this trait in the existing breeding stock, but we can use gene editing to, once we've done kind of the basic research to identify this gene is related to this trait, you can take, you can use gene editing to make a parent variety of hop that has that trait, and then do the cross to develop all those offspring. And hopefully you have this new trait incorporated that wasn't really available before in the natural germplasm stock. So in that case, gene editing can come either before the breeding pipeline or after it.
Yeah, interesting. And I don't want to lose this thread too, because you mentioned something, or you mentioned agrobacterium earlier, and that's really, really interesting, right? Because this is a piece of how you actually get the genes into the hops, right? We've talked about, some ways where we might think about inputting or using gene editing, but you still have to actually physically transfer the material into the hops. Now, you mentioned agrobacterium, and that's kind of your vehicle for delivering the blueprints, but I had a couple questions about that. So first, one is how did you identify agrobacterium or as a suitable deliverer of blueprints, right? I mean, I can only imagine that had to take a long time for somebody to figure out that this could actually work.
Yeah, so this is a really fascinating story for me. So agrobacterium is with the caveat that this is going to sound a little bad at first. Agrobacterium in its natural agricultural context is normally a plant pest. It is an organism that it causes what's called crown gall disease in its natural context, which doesn't affect hop all too bad, although it is known from hop, but it's more problematic in kind of nursery crops. Certain flowers are really susceptible to it. At any rate, it is really unique among plant pathogens in that it causes disease by delivering DNA from its cell into the plant cell, and the plant cell will stably express that transferred DNA and make a niche for agrobacterium to colonize and make a specific food source for agrobacterium that only it can metabolize. So a really, really clever evolutionary strategy. And so people have been studying agrobacterium since the late 1800s. It's been kind of real interest. So it's very like wide host range as well. I think there's something like at least over 1,000 species right now that are known to like be susceptible to agrobacterium. And so we've studied the genes that actually do get injected into the plant to cause the disease. And somewhere about the early 80s, researchers figured out that there's the specific genes that get transferred into plants, which we call transferred DNA or tDNA. And we figured out kind of where the borders of that are within the agrobacterium's genome. And we figured out how to delete the genes in agrobacterium that cause the disease but still leave all the machinery that it uses to transfer the DNA in there. So essentially what we have done with agrobacterium since the early to mid 80s is to create versions of agrobacterium that are what we call disarmed, so they don't cause any disease symptoms anymore. And then we can put our own genes of interest into the agrobacterium, hand it off as if, this is the natural, the genes that they would transfer in their natural context. So they now don't transfer these disease-causing genes, but now they do they work for us now.
Right, and that was actually going to be my next question. Is like, okay, agrobacterium. I mean, if it's causing gall and these diseases, it sounds like you've deleted the bad genes out of it. Well, I'm going to say bad as like a loose term or an unscientific term, but you've deleted the disease-causing genes out of it. But what happens to the agrobacterium? I mean, where does it go whenever you deliver these cells to the hops? Is it still there, or is it like, are all gene edited hops infected by agrobacterium, or like what's the?
Well, so when we do transformation in a tissue culture context, we want the agrobacterium to grow in culture with the plants for a little bit, because we want to have as much surface interaction with them as possible, have the best chance of delivering genes into the most cells. But then, when we want the plants to then grow and replicate, we want to kill off the agrobacterium. It's not needed anymore. So because we've taken away the disease-causing ability of the bacterium, it's not going to persist as easily as it would in a natural context. And we can actually solve this problem usually pretty easy by washing the plants off with sterile water and applying a light antibiotic that affects the agrobacterium, but not the plants.
Interesting. That makes total sense, right? Wash the plant off, wash your hands and use antibiotics, right? or soaps and stuff. That's kind of what we talk about often.
And one other thing that I wanted to kind of point out about agrobacterium is it is usually thought of as as a plant pest, but there's a really interesting finding from a couple of years ago, which does have implications for hop. So hop and several other plants such as grape have been interacting with agrobacterium through evolutionary history. So they've been kind of interacting with each other for millions of years. And once we got a really good sequence of the hop genome, there's a group that found there are genes from agrobacterium that we ordinarily think of as disease-causing genes that persist in plant genomes. So at some point, a natural gene delivery event happened in hop's evolutionary history, and it still retains those genes, and those genes are still turned on, which kind of implies that that this must have had some kind of positive effect on hop's evolution. And so, one argument you maybe could make for using agrobacterium to deliver genes from outside the plant, that's not natural. That's not something that happens in nature. You're doing it in a laboratory. But I think that kind of demonstrates that actually this process is natural. It's been happening for millions of years. And what we're doing actually isn't really fundamentally different.
Right. We're just kind of calling it a different name, or maybe we understand it more now, right? When we understand it, we're able to use it and control it. We have some tools available to us now to be able to do this. Yeah. I think that's really interesting. That's sort of a broader discussion about like GMOs and genetic modification and all that stuff in general. But I'm not going to open that bag of worms right now, because I do want to get to some of the OSU projects that you guys have proposals out for. Or these don't have to be just OSU projects. I mean, these can be projects that you're interested in studying as well. I know you have some funding proposals out there for some things that might be interesting to look at. But this is kind of what I'm looking here for, is just what are you interested in studying with how do you plan to use gene editing to change or assist hops?
So as I mentioned, disease resistance is a really big one. I think that's really important, especially for hop growers, because powdery mildew in particular is a pretty prevalent disease of hop, and there are, cultural methods of controlling powdery mildew, which often involve to some extent applying fungicides. But kind of the best method we have of resisting it is having plants that just have this genetic source of resistance to powdery mildew. And like I mentioned, the project that we originally started this work with, what sought to identify this resistance gene, that project has turned out to be a lot more complex than we initially thought. We're going to need a little bit more genome sequencing from varieties that actually have this, that or actually display this source of resistance as a trait, in order to kind of figure that out. But I wanted to mention that Michelle Wiseman, who's a PhD candidate who works with Dave Gent at USDA, she's, first of all, like right after I got to OSU, she had just started as a PhD student at that point, and she, I think she contacted me before I got to OSU, actually. And she has been interested in biotechnology for a really long time and she wanted to work closely together on developing these gene editing tools. And so her primary study, I'm hesitant to talk too much about her work, but I would definitely recommend you having her on at some point. She's a really bright student.
Yeah.
But she is working also on powdery mildew resistance, but she's working with a different family of genes that are called MLOs, mildew locus O. And these genes are actually what's known as susceptibility genes. So they're a gene that the plant has that powdery mildew manipulates in order to gain entry into the plant and infect it. And in many other plant species, so this is kind of a universal plant family of genes. And in many other plant species, mutations to these genes actually make resistance. So in this case, you could turn off a specific member of this family and then confer resistance to powdery mildew. And it has actually a lot of advantages over resistance genes, in that a lot of times with resistance genes, breeders can add a resistance gene to new varieties, and then the pathogen just kind of evolves around that. So the resistance genes don't work anymore.
Yeah.
With these susceptibility gene conferred resistances, in other crops that have used this MLO strategy, it's been a pretty durable form of resistance. So it's kind of wide ranging, and then a lot of different strains of the powdery mildew pathogen are affected by them, and they don't easily evolve a way around that trait. So I think MLOs are really, really promising tool for powdery mildew resistance in hops. And Michelle's work has she's done a lot of work to identify candidate MLOs, and now, since we've worked on developing gene editing methods, she's planning on using and is actively doing experiments to use gene editing to turn off certain MLO candidate genes and see if those will kind of confer the same thing in hop that they have in other crop species.
Wow, cool. It's kind of working at it from two sides, right? Increasing resistance is kind of what you were looking at in terms of identifying genes that increase that resistance, but then she's looking at reducing susceptibility. So
Yes.
if you can, and think about the power there too, right? If you're able to combine both of those, right, by both increasing resistance and reducing susceptibility, then may really, really provide some substantial benefits to hop growers. And that would be great, right? Because one of the big issues for powdery mildew is it increases fungicides. Fungicide use, it is a big deal for growers. That's an increased cost. So, all those things, it's something that growers have to manage. And then also, my understanding too is it increases or decreases yield somewhat too. So it's a very...
Well, then there's the regulatory barriers. So some countries have more specific or more stringent regulations on how much fungicide residue you can have on it, and it kind of hurts hop exports to like the EU is a good example.
Yeah, exactly. Where and in places where powdery mildew isn't necessarily as big of a problem, right? Where it is here, there are parts of the world where powdery mildew isn't as big of an issue. So
That's
yeah,
right.
that's fascinating, right? So just this, so using gene editing for this very targeted thing can have wide ranging consequences for hop growers and thus brewers by extension. That's pretty cool and cool to think about, right? That's like a supply chain issue. There's also some very specific hop characteristics that you guys have got proposals out there, and these ones might be sort of more interesting or more directly applicable to brewers. In that, I think you have a proposal out there for for dealing with alpha acids, and then also for terpenes.
So we have proposals for the alpha acid one, and then we're looking at xanthocumol, which is, if people don't know, is a phytopharmaceutical compound from hop that I think it's kind of has a lot of interesting putative biological uses. That antioxidant, has some promises as an anti-cancer agent, but I think what has the most evidence for right now is treatment of metabolic syndrome, fatty liver disease in humans, and what is it called?
IBS.
Irritable bowel disease.
IBS, IBS or
yeah.
IBD. I can't remember. Yeah, exactly. One of those. I can't remember exactly which one it's called, but yeah.
So basically, xanthocumol is only synthesized by hop. It's a very unique compound, but it's not present at very high levels in hop. So I think right now it's very expensive to extract, and I think how it's extracted at the moment is high alpha varieties, where an extraction is done for alpha. They'll take like the can't remember what it's called, but the spent hop material that's already been extracted, do a secondary extraction with a different solvent to get xanthocumol, and I think just the cost of that procedure ends up being too expensive to use at the levels you would need it as like a health supplement. And so our work is looking at, I think, 7 or 8 genes we identified that either knocking them out or boosting them up might boost the levels of xanthocumol. So it's just kind of a basic research project to see which of these genes are going to have the biggest effect, and then hopefully can we combine different edits of these genes to produce a hop variety that can make a lot of xanthocumol and make it kind of marketable. So the alpha acid project, we have a grant with or a pending proposal with HRC to do that research, and that one would be just kind of a pure basic research proposal again, where it's kind of following up on a story from I don't know, the last 10 or so years, where people have done a lot of identification of the enzymes involved in producing alpha and beta acids. And we know almost all of them, except for at the very end of that pathway where those compounds are produced, you basically end up with this precursor compound that branches. And on one side of the branch, you get alpha acid, one side of the branch, you get beta acid. And the enzyme that catalyzes that transition from that precursor compound to alpha has never been conclusively identified. But there were a couple of sequences for this. So the gene would be called humulone synthase. Humulones are alpha acids. There are a couple of sequences that were deposited by this group, I think back in 2014 or something. And so these researchers are no longer working in hop biochemistry area, but I was able to follow up with them and kind of hear the story of how they got to those sequences. And basically their project just kind of got interrupted, and they had a pretty high confidence that these were the alpha acid producing enzymes, but they were never able to kind of conclusively prove that and write it up into a paper that was that reviewers wouldn't be questioning. So it kind of just got dropped. And so I think that there was enough evidence there to say, okay, instead of going at this from kind of a conventional enzymology biochemistry perspective, what if we take these genes that they've kind of, given us a candidates for, and knock them out with gene editing, grow up the cones and see, have we now eliminated alpha acids? And so, that wouldn't result in a super useful variety. I guess the expected phenotype is a 0 alpha variety, which, there are conventionally bred varieties like that out there. But what that would do is just kind of give us, okay, now we've conclusively identified this enzyme in alpha acid biosynthesis. Can we look in the natural germplasm for varieties that have really productive versions of genes that code for this enzyme? And can we use that in breeding to make super high alpha varieties in excess of what we already have?
Exactly, right? That whole tautology, the whole circle that comes back, right? You do the basic research to identify the genes that cause alpha acid production in hops, and then you can use those genes in breeding practices and look through the current germplasm and see which ones could we use to make high alpha acid hop varieties. I mean, and we could be much higher than what we're using, than what we're even getting currently. That's a pretty cool application. And then, of course, the health impact of xanthohumol as well, right? I mean, that's a pretty cool one too. If you're able to side stream some hops, I mean, especially as we're sitting right now in a hop, where there's a lot of hops sitting in the in storage, there's potentially a good opportunity there for hops as a secondary market, not just going into beer.
Yeah, and I would say on the terpene synthase thing, we don't have any active grant proposal for that right now, but that's something that I've pretty much been interested since the beginning, where my father-in-law was talking about making a custom variety of hop. Is there are these enzymes called terpene synthases that make, terpenes, which are the kind of volatile chemicals that give hop its unique aroma, along with thiols are another compound that influences that. But terpenes, there are a lot of terpenes, and they each kind of have a different note that they'll impart into aroma or flavor. And I think an interesting thing to be able to do would be to use gene editing to knock out individual terpene synthase genes, so you can eliminate one terpene or boost it, or get new combinations of terpenes, and just kind of get altered profiles of hops, which from my understanding, a lot of times that's really what brewers are looking for, is just kind of novelty of varieties, right?
Right.
So I think even if you went in blind, kind of editing these different enzymes and didn't really know what, you can't really predict what's the specific flavor outcome. But if you just generated new varieties, that might be a way of generating novelty, while using gene editing to do it to maintain good agronomic characteristics.
Wow, interesting. Yeah, so we could actually, breed new aroma hops, I guess, or unique varieties, right, by changing these terpenes. And that's actually really interesting, too, because that's kind of one of the yeast ones. Berkeley yeast, that's how they got their start, was using terpenes, I think, what was it from? Was it from mint or from basil? Or
I think
something
both.
Yeah,
so they had a geraniol from basil and something else from mint. I can't remember. Linalool,
maybe. Yeah, exactly. That's how. But the yeast make terpene synthesis too. So they just upregulated those things in yeast, inserted that DNA, and boom, yeast can now produce terpenes and increase that flavor characteristic. So it's really interesting. And so that kind of is, this is one of those things. I mean, we didn't leave ourselves too much time to talk about the future and far future of gene editing hops, but this is certainly one of those things that could be done in the next few years or the next decade or whatever we're looking at, right? I mean, the xanthohumol, the alpha acids, the terpene synthase, all of that stuff. But we do have to ask one question before we sort of round things up, and it's that regulatory framework. So are gene-edited hops allowed in the United States?
So short answer is yes. Long answer is this is very complicated. So I think a lot of researchers who work in academia, just kind of using this as a discovery tool, tend not to verse themselves in kind of the regulatory framework here. And I would absolutely include myself in that until about, 6 months or so ago. I've been kind of trying to understand the kind of regulatory pathway to getting something approved to be grown in the field. And so one element to this is the USDA APHIS is the name of the organization that regulates whether we can grow these biotechnology manipulated plants in the field. And basically, so they have adopted a new regulatory framework for this in 2020 that was called the Secure Rule. And the guiding philosophy of this is basically, if you create a modification with gene editing that would be even theoretically achievable with conventional breeding, it should be regulated as if it were a conventionally bred one. Because we think really how they have in the past evaluated plants has been, what tools did you use to do this? What did you use, agrobacterium, or did you use conventional breeding? But I think they've kind of gone towards what
did you actually
do? These are just processes, and the final product is what matters, regardless of how you got there. So if anything that has been gene edited in a way that you could do with conventional breeding is produced, technically you're allowed to just put it on the market. It's not subject to any regulation. Just like, if you you cross varieties of tomatoes in your garden and produce seeds, that wouldn't be regulated. For anything that contains DNA that has been delivered that would not be possible for conventional breeding, there's this kind of second step of a process where you have to submit what's called a regulatory status review request, which is kind of a document where you outline exactly what is in every part of the DNA that's been inserted. And then the regulators will make a determination as to whether they think this presents what they call a plant pest risk. So that is really the only criteria that USDA uses to say that, you can't grow this because we need more environmental studies, basically. And this might relate to things like if you wanted to do, what we've done in a lot of corn and soybean that's grown in the U.S., like herbicide resistance or incorporated insecticides. Those are definitely something they're going to want to take a look at and do further studies on. But I don't anticipate things like altered xanthohue multipresent a plant pest risk, according to them. Now, a caveat there is because this is all really new and regulators really kind of haven't figured this out yet. The EPA and the FDA also claim some regulatory overview of potentially genetically modified crops, and it's kind of less clear how they are going to view specific traits. So EPA, was set up to regulate pesticides, basically. So they recently put out a thing where they are going to want to look at anything that modifies the plant's growth characteristics, which that might include, if you wanted to alter the height of the hop or make it more branchy, they might be interested in that. They would definitely be interested in at least looking at something that, would alter the powdery mildew susceptibility. But basically, we don't know yet how easy gene-edited plants are going to be to get through this regulatory process. And it's going to just be a trial and error of submitting these plants and kind of dialoguing with these regulatory agencies. And this is not just hop. This kind of goes for all crops besides the major commodities, where big agricultural companies like Bayer and Corteva have kind of worked this out with regulators already. But I think there's kind of still a lot unknown as to the regulatory side of how easy it's going to be. But in general, the question of can you grow them? Well, yes, in some cases, but it's going to be a case by case basis.
Right. As with so much, it depends.
Yes.
And then I think that's a good place to leave it too with the regulatory topic. So I always ask this question towards the end of the show. If you want brewers to take away just one thing from today's episode, what would it be?
So I would say, I guess to think about specific traits that brewers would be interested in that gene editing might be able to address. So like you brought up hop creep earlier. I was actually, I think just yesterday or the day before, emailing with Laura Burns about a upcoming talk we might be doing together, and she mentioned hop creep as something I could discuss. And I had not really looked into hop creep at all before. It was news to me that that you said earlier specific genes associated with it in hop had been identified
with amylase production. So up to that point, there were no specific genes that were identified for amylase production. So that amylase could create hop creep. There's one more step between there, but still, there's a lot to look at. Yeah.
Just the fact that there are specific, like, that work being done would be a prerequisite to using gene editing in the first place.
Right. Yeah.
But I guess my point there is I have interacted a lot with hop growers, not in the hop merchants, and I kind of know what traits they're most interested in and would be of most benefit to them, whether just from a basic research perspective or thinking about in the longer term, can we use these in the field? But I don't feel like I've had as good of a feel with what properties are brewers most interested in. So I guess I would say I would love to hear from brewers on any additional ideas that they have for what this technology could be used for. Hop creep being a really good example.
Yeah.
So I don't know if my email is going to get put in the show notes or anything, but
sure, I'd be happy to share it with anybody that wants it if you're interested.
I guess my email is just chris.willig@oregonstate.edu. And then I will probably be at the Craft Brewers Conference in 2024 in Las Vegas. So maybe come up to me there if you want to talk or have any additional ideas.
Well, and I'm sure Jordan and I will spend some time on next week's Applying the Science episode coming up with some of our own ideas, just probably mine and Jordan's. But yeah, if you're listening to the show and you've got an idea, send me an email and I'll send it on to Chris. And so that's really cool, right? To think about the future of hop breeding and using gene editing technology. So what are brewers interested in? And what are also growers? Well, cool. Chris, thank you so much for joining me in the Brew Lab today and talking about this really cool and really new topic.
Yeah. Thanks for giving me the exposure too. I really think it's important to kind of dialogue with the interested communities of what my research will affect. So I think this is a really good opportunity for doing that.
Well, absolutely. There's certainly an interested community here of listeners that listen to this podcast. I think this is certainly going to be right up their alley. So thanks again. And of course, next week, Jordan and I will be back applying the science of this episode. So we'll see y'all then. The Brew Lab is a production of Brewlosophy, where they who drink beer think beer. Don't forget to visit brewlosophy.com to read about our weekly experiments and other brewing adventures, and listen to us talk about it on our other show, the Brewlosophy Podcast. Thanks to all of our sponsors and patrons that help make this show possible. If you'd like to receive a reward for helping us do what we do, visit patreon.com/brewlosophy to see how you can do just that. Thanks so much for listening. We'll be back in the Brew Lab with another guest next week. Until then, think beer.